Welcome to everyone for joining this webinar. We are going to have a seminar on the topic of RNAseq supported Mendelian Disease Diagnostics, and we have two speakers today. It's Vicente Yépez and Christian Mertes. And before handing over to our speakers, I'd like to say a few words around GHGA. So we are the German Human Genome-Phenome Archive, and our mission is to provide human omics data sharing in a GDPR-compliant manner. That means we'd like to provide clinical omics data and make them available for secondary use in research. And hopefully that will then lead to translation of research insights and also improve genomic medicine. And we're not doing this alone. So there is a diverse community involved in this. We are computational biologists, molecular geneticists, clinicians, physicians, and also patients. I would like to introduce our speakers today. First up is going to be Christian Mertes, who did his master's and his bachelor in bioinformatics in Munich, and did then a stint at Stanford in the US before completing his PhD here in Munich in the Department of Computational Molecular Medicine. He has and still is developing statistical methods to identify RNA outliers, so he's still working in the RNA field, and he's also the main responsible person to set up the faculty IT infrastructure. And for GHGA, Chris is the workflow workstream coordinator working on standardization and harmonization of sequencing data, and he's also the deputy speaker of the Bavarian Cloud for Health Research. And the second part will then be presented by Vicente Yépez, who did his bachelor in industrial engineering in Mexico, went on to do a master's in mathematical modeling and engineering, and then a PhD in bioinformatics at TUM, where he's still working as a researcher and a scientific manager. And he is also the co-lead of the European Solve-RD RNA-seq analysis working group. So with the background of both of them, I think we are in very safe hands. And with that, I also hand over to Chris.
Perfect. So thanks a lot for the invitation and that kind introduction. And so we will talk today about RNA sequencing supported Mendelian disease diagnostics, basically in two parts about robust outlier detection methods and also the application in the on disease cohorts. So what will we learn today? Basically, a first introduction on rare diseases and why do we study them, what is interesting about that. And then, as I said, we will dive into the tools and workflows for RNA sequencing-based diagnostics. And so here we have the gene expression outlier detection or splicing outlier detection and this full end-to-end analysis workflow. And in the second part, Vicente will talk about the application of it in a mitochondrial disease cohort, but also different approaches between like RNA first or DNA first approach, and of course, the analysis in the Solve-RD project. What are rare diseases? Rare diseases are defined as life-threatening or chronically debilitating, and usually have a prevalence of 1 in 2,000. Depending on where you live, the EU or the US have different definitions, but it always boils down to 1 in 2,000, more or less. Interesting also for the rare diseases is that 80% of the rare diseases are genetic or of genetic origin. And that means that they are usually caused by variants in a single gene, and they are usually inherited. And so why are we looking into that if they're so rare? The thing is, with rare diseases, currently the estimates are that 4 million people in Germany are actually affected by rare disease and living with it. And worldwide, that estimates goes to like 350 to 400 million people. And so each disease is maybe rare, but collectively, rare diseases are actually not rare and affect around 5% of the population. So how do we help those rare disease patients? Currently, the first tier of diagnostics is DNA-based. And so here what you do is you usually take blood. You do then your sequencing. You can do candidate genes or panels, whole exome, or the whole genome sequencing. If you have that, then you basically look into the variances and follow the ACMG guidelines, which are the American College of Medical Genetics guidelines on how to interpret those variances. If you filter them down to the most potential disease-causing ones, you usually have like 10 or 2 of the variances, and those variants then you basically look into manually and overlay them with with phenotype or other omics data to kind of like get your functional validation of your variant. But the problem is that most of those variants are still in this WUS category, which is like the variants of uncertain significance, so that the clinicians cannot really make a conclusive diagnosis out of it. And so currently in rare disease, because it's so rare and so hard to diagnose, actually such a diagnosis odyssey takes almost 5 years in average, and only in 50% of the cases actually successful. So that means half of the population are still left without a diagnosis or a molecular basis for their disease. And so we actually do have to need to look beyond the DNA and have to figure out other ways of doing that. And that's kind of like where we are with the DNA, with the RNA. So taking the RNA is basically functional the readout of the DNA. And so can you assess the predictable or unpredictable effects of those variances? So here you have a gene model. And that gene model, you have those exons. And in the exons, we are pretty well, like we understand the effect of those variants, if they're like missense mutations or stop codons, so that we understand. But actually everything around it, if it's in the enhancer or promoter region, somewhere in the intron or around the splice site, it's very hard to actually predict the effect of it. And so that's why RNA sequencing is needed actually to interpret those variances and the effect. And so all those different kinds of variances we see here, which are depicted, can actually then be, they show the effect on the aberrant gene expression or aberrant splicing, or even on monoallelic expression. So how does the whole workflow looks like? So actually RNA sequencing is really complementary because we look at the effect and not the variant itself. And so it's really a complementary tool in diagnostics. And so what we do usually is you have your whole genome sequencing. If it's inconclusive, you take the patient cell lines, you do the sequencing, then you do the outlier detection, so bioinformatics, and then based on those information, you do the RNA-seq supported diagnostics. In the first part, we will focus on the outlier detection and the methods, and then later on the diagnostics. Yes, so if you look into the tools, if you have RNA sequencing, the first thing usually what you have to do is really to correct for sample covariation in the data. And so when you look at the data, you can have effects of biological things like sex, age, general health of the patient, but also actually technical things or the origin of the patient. And so the different papers here, Brechtman et al. And Fraser et al., they kind of use different tools on actually doing the correction for sample correlation. Another important point is that with rare diseases, we are not comparing 2 different conditions, but actually we want to have one outlier in the population. And so we are looking here for aberrant expression and not differential expression. But you can see here on the left side, the first part is the normal thing you know from DE-seq or HR. You have 2 groups, A and B, and then you basically compare them. But what we have is rather we have like a huge population, and within that population, one of the gene is basically, or one of the patients basically driving out of that population, and that thing we have to detect. So it's kind of like the outlier is the signal in rare diseases. Yes. So diving a bit more into the correction of the samples, and so we developed over the past years together with Felix Brechtman a tool to use an autoencoder. And so here we do actually a context-dependent outlier detection. And what you are starting usually is with the raw counts, and you have this matrix, then you usually have this batch effect inside the count matrix. And if you look closely, you have on the lower part on the left side, you actually have one sample which is kind of like dropping out of the population. But if you look at across the whole thing, it would actually not really pop out. So what we need, or what you want to do, is we actually run an autoencoder to model the data. And then when you have the model data, we can contrast it. And by doing that, we can have controlled counts and actually find this outlier. More specifically, when you take one single gene, on the left side, you have the distribution without the corrected data, so it's just the raw data. But if you now correct the data on the right side, we can see that the single event is basically driving out of the population and is detectable. To give you a hint what is like an autoencoder, so we use here denoting autoencoder to control for this known and unknown confounders. And so it actually comes from the imaging field. So where we take an image and where we want to basically make a classifier and figure out what that image is and basically say to remove all the noise in there. So we have this input, that's X, that's our input. We put a lot of noise on it, so we corrupt the data. And now the question is, can our autoencoder actually figure out where the numbers are and what the noise in the matrix is? And so having a function to do that, you can see that we basically can recover the image correctly. And that's the same thing what we also do with the RNA sequencing data. We basically try to remove all the noise from the signal. And then by contrasting that, we can then figure out what the outlier is. So having now basically those outliers, the question is, how do we model the data? Till now, usually the people use set scores, where you basically figure out how far it is from the standard deviation. But in RNA sequencing data, we actually deal with count data, with discrete count data. And so it's always more advisable to actually reuse the proper metric distribution to detect your outliers. And so here we use the negative binomial distribution to detect the gene expression outlier. You have now two examples on the left side and the right side, like with the normal expression. You can look at the QQ plot on the top, where you really like underline and model really well. On the bottom, you have the normalized counts of the population. On the right side, you see an outlier example where you still have the majority of the data is modeled pretty well, the diagonal, but then only you have just 2 or 3 outliers which are really popping out of the population. So here we can prove that we actually can model the data with a negative binomial distribution. To validate actually that using now the autoencoder to model the co-founders in the data, and then also using negative binomial distributions, we wanted to figure out actually and revalidate that what we are doing is the right thing. So what we can do is we can look into a huge cohort, which is T-Tex, which is a bigger cohort of healthy people, but in the population, you have usually red variances, which are not disease-causing, but still affect the gene expression. And so the assumption is that if you have a variant somewhere in the gene that affects maybe like a transcription factor, and then the transcription factor is not expressed in the gene, so it basically removes the thing. We can look in T-Tex in our 900 samples for rare variances, and then basically predict, see if we find the outliers in the RNA. And doing that, we can now kind of have a benchmark and can compare against other methods like PCA or peer correction. And as you can see, as running Outrider, we have a high enrichment of those rare disease variances and the outlier. So a quick take home for the expression outlier detection is that, yeah, autoencoders are helpful to control for co-founders. It's important to reuse the proper distribution for your count data. So here, negative binomial, and it's basically outperforming the significance-based Z-score outlier detection. And we have a nice package on Bioconductor where you can basically run it with up to 3 lines of code to actually run your analysis pipeline. So the next step was kind of like obvious. We have the expression outliers, but as we know on RNA, we also can look into splicing. And so we can take the same idea from the expression where basically finding outliers is an autoencoder, we basically approached this splicing part. That was driven by Ines Scheller from the lab. So here in splicing, it's a bit more complex because with gene expression, we just have one value. We have the full expression of one gene, and that's just one value. With splicing, as you can see, we have a lot of different ways of splicing. And so really figuring out which intron is important, and that's actually more a ratio here because we compare one intron against the others and the usage of the intron. So it's harder to actually model that. And so here, what you see here is an example of one of our first cases where we basically find an outlier in our data. And so here we have a single variant in the intron somewhere like in the middle. And that intron actually creates a new cryptic exon. And as you can see in the two patients in the upper part, basically you have the higher usage of the cryptic exon compared to the others. Yes, so at the time when we developed the tool, there's a lot of different ways out there, but each of those tools have kind of their limitations. And so one thing is that this, as you saw in the example, it's very important in splicing that you actually are annotation free, and that you can basically look at any data in your RNA sequencing data. Because if you are only bound to your annotated data, you might actually miss those rare events which are outside the annotation. Also, as you know, the same thing applies for splicing, is that we actually do have to control for covariation. And as you can see here, we have Cummings et al., which one was also the first papers, which kind of like just used the annotation and just counted. So they actually do just a cutoff-based approach. The next one was Leafcutter, which was used by Kramer et al. So it was more kind of like a D-seq approach where we had one against the rest. So we did count for count data distribution and did a significance test, but all the control of confounders or annotation free was basically not there. And so over the years, also other two tools came, but always had some limitations here. And so that's why we developed Fraser. And actually currently in press, we have a second version of it, where we improved the metric of detecting aberrant splicing. So what is especially the improvement in Fraser 2 is that we actually have a novel intronic centric metric, and we call it the intron Jacquard index. I will give you a quick introduction to that. As you can see, it's like splicing is complex. And so we wanted to really have a robust estimate for a specific event. And so what you can see here is the yellow part. That's your intron of interest. And we want to figure out, is that intron differentially spliced or not compared to the other samples. And what you can see here on the red side, we can have all the reads which are coming through the donor. And so we can have split reads which are spanning to a different intron. So here you can see they go far to the right, or even the reads which are just spanning the donor side and are not split at all. The same way, you can also look at the acceptor side of your intron of interest. And so here you have reads which are basically split reads going backwards and to the left side, and basically get their splice there. And also, again, the reads which are not spliced at the acceptor side. And so we can now take the union of all those reads which are around that splice side or affecting the splice side, and then dividing it by the numbers, by the reads of that splice side actually. And so having that, we have a really robust estimate across also the bigger vicinity, and can use that for modeling the splicing effect. Here for splicing, we basically took the same approach as in gene expression. So we have our input matrix, we do a denoising autoencoder, where we basically model data into a hidden space. And then from the hidden space, we do again the prediction for the controlled data. And by contrasting again this controlled data, we can then actually figure out our variant of interest, sample of interest. Doing that in real life, in GTEx, you can see here before the corrections, you have a huge batch effect in the cohort. but running the autoencoder, we can actually remove the batch effects. That's on one tissue, and what you can do now is we can actually do it across all the tissues in t-tags. And as you can see here, if it's the red bars are basically the data for uncontrolled data, and it's always higher. And if you basically run our autoencoder on the green part, then we reduce the correlation between the samples, which is important for the outlier detection. As the same with Outrider, we wanted to make sure that we have the proper splicing detection. And so here, since we are looking at a ratio between one intron, all the reads in the intron versus all the rest, we have here, we use the better binomial model. And as you can see on the right side again, we can nicely model it. And so the QQ plot is more conservative a bit, but still is modeling the right thing. On top with the splicing, we also figured out that it's important to have actually kind of like a biological relevance score. And so here we use the difference between the event to the population, which is called the delta Jacquard value. And it kind of tells you how far is that away from the population. Meaning if you have like 30% missplicing, that would mean here that Jacquard is 0.3 away, meaning 30% missplicing happening. Also, again, with Fraser, we did a similar benchmarking to prove that the method is actually detecting something biologically relevant. And so we again looked at all the splice variances in t-tags in the cohort, and then figured out if we find actually for those splice variances also an splicing outlier. And here we use the different tools to actually validate it. And as you can see, is the highest curve is Fraser 2.0. Where we have the highest enrichment for splice variants. You have here four different models. So we use different tools actually to predict splicing variances. The first one is like the default using VP, the annotation where we just look at the splice site variants, so like two bases into the intron, which are very prominent to cause splicing. But we can also use models that predict actually splicing, so MMSplice, Splice AI, or the ABSplice. Okay, so having now the tools out there, what is also important is actually that we look at QC values of the RNA. And so as in the beginning, we always have, like for diagnostics, a complementary thing. So we have to make sure that the RNA we are looking at is also actually the same from the DNA. So we want to actually compare, like we can use some metrics there to actually compare the DNA with the RNA. But also it's important to really see the sequencing depth of the sample. So if you have actually an outlier here in the sequencing protocol. Often also we have some sample swaps. So it's important to really understand that we have the female or the male sample in here. So that's actually a very easy QC step. We just like look at the expression of the Y or X chromosome. As I also said earlier, we have to do this matching between DNA and RNA, so we can actually use the variants, like common variances in the data, and then correlate them and see if that samples actually belong to the DNA or not. Interesting is also to really see that if you look into the tissues, because the tissues are very different and the expression splicing is different in the tissues. So we want to also make sure that the variances, the RNA samples we are grouping are actually belonging to the same tissue. And so we can do some estimates based on the expression profile of the sample to which of the tissues that sample belongs. And so you can see on 0.4 that is really well correlates with the data. Yes, so take home here from splicing. So again, it's very important that we are annotation free so that we can detect novel and cryptic events. It's very even splice site centric metrics exhibit a strong covariation. And so we have an intron Jacquard index introduced, which is really a robust metric to detect aberrant splicing. And again, here we have a tool in Bioconductor, which you basically can use out of the box to run your diagnostics on it. So to wrap up, so you heard about all the different tools we have, but those are very much driven by the bioinformatics community. And for clinicians in the diagnostics, it's not really helpful because they cannot rerun the tools. So what they need is actually a complete end-to-end pipeline where you have from the sequencing data down to the visualization and integration of the data. And so over the years, Vicente developed a drop pipeline to detect RNA sequencing outlier in RNA. And so here it's really a full pipeline, and that's also actually part of the GHGA development in the workflow stream. And so it enables the researcher and clinicians to actually run a full analysis in 2 days from the sequencing data, and then visualize it, give it back to the clinicians so that they can browse through in the web browser to the report. Okay. Yes, so that was the first part, more the technical side. So we will have now a short break. So you have the opportunity to ask some questions. And if the questions are over, we would then go into the diagnostics part. Any questions? So also if you are shy, they are not recorded, so they will be cut out before we put it online. So don't worry to ask some questions. Then I would say, Vicente, you can share your screen or get ready already.
Yeah, so then I guess I'll start with my part then, and then maybe we'll have questions at the end. So I guess you can see my screen now. Thanks, Chris, for the first part. I'll now resume exactly where you left us. So indeed, we heard already from Chris a bit of the method, so what comes before, and now we'll get into the maybe more interesting part. So you do something with a goal, and now that goal is to eventually diagnose patients. So how do we do this result interpretation to reach that? So what is needed is this integrative analysis in which we analyze the RNA not alone, because the RNA alone is very difficult to reach a diagnosis just with it. So we analyze it with the DNA and with the clinical reports. But that's not the only thing that I mean by integrative, and I find it very important to also combine together the bioinformatician part with the medical part and with the experimentalists. And working together, it's way faster and more precise. We'll get a diagnostics from experience. So this has been done already throughout the years since 2017 from the pioneering studies that we had here in Munich and at the same time at the Broad Institute in the US. And basically they do this to detect this aberrant expression, splicing monolithic expression, and combined with the information from DNA, either from whole exome, whole genome sequencing, we have had some diagnosis uplift or from 8 to 35% in different, taking different tissues, blood, muscle, and skin usually, and in different rare disorders. And now in this year, we have had a actually great uplift in number of studies, and now it's really becoming worldwide. It's not just exclusive to US, Canada, and Germany as before, but now we have studies from Hong Kong, Netherlands, Australia, and UK. So it's definitely getting popularity, this approach. And basically, we have two different ways to detecting outliers. One is what we call the RNA first, in which all expressed genes and junctions are tested. So now we're talking about the tens or hundreds of thousands of features that are tested, which makes multiple testing correction quite harsh. While many times in diagnostics, in rare disease diagnostics, we have a set of candidate genes that we're interested. For example, if we test even all omnium genes, we're just in the 5,000, or some patients we already know the disease they most likely have, so then that can restrict to the hundreds. If we test already genes that have that are known to have already high impact, we're just 10, 20s, or other candidates we could be really 2, 3. So we reduce a lot the search space. We have adapted our methods to do that, both Fraser and Outrider. And indeed, for example, in the blood cohort from SOFAR-D, which is one of the biggest ones that we have, we see that indeed it's complementary to focus on certain genes than with respect to testing all, but of course, testing all will give even more results in total. So we have worked throughout the years with different data sets from different tissues, from different disorders, for example, ALS, mitochondrial, maybe the one that we have studied the most here in Munich, the UDN, which is the Undiagnosed Disease Network, mostly of US, and lately, since last year, with the SOFAR-D as well. And you see it's usually hundreds of samples, and our methods, both Outrider and Fraser, they return just a handful. Maybe Outrider is around 5 outliers per sample, and Fraser is usually less than 20. So it's not a lot to inspect. Especially if you add this to phenotypic and genetic information, you end up with very few, 2-3 cases to inspect per sample. So why do we do this? So why detecting even aberrant gene expression? So we have this gene that is very much reduced in a specific sample with respect to a population, because it can be indicative of variants. It can be indicative of variants even before the gene, in enhancer promoters, in the UTRs. It can be indicative of large deletions. This has happened many times, that the deletions were missed by a standard whole exome sequencing analysis, and through finding aberrant expression, we can find them. Or it can be due to intronic variants that they activated a new this exon contain a premature stop count, and that led to aberrant expression. So I'll present now some cases. I think five in total, real life cases that happened to patients where RNA was directly useful. For example, this one, a patient from Japan, from this Chiba University Hospital that we were a couple years ago, suffers from a mitochondrial disorder. Whole exome sequencing revealed a heterozygous missense, probably damaging variant in NFU, which is a well-studied or well-characterized mitochondrial disease gene. Here is the variant, the missense variant that was found by whole exome sequencing. Now this gene is recessive, so that means we need variants in both alleles. So this, even though it's very promising, is not enough to diagnose. Then what we find with RNA, it was a 50% reduced expression. And a missense variant usually doesn't cause reduced expression. Moreover, we found that the missense variant here detected by whole exome sequencing was monoallelic expressed. That means that there's something going on in the second allele that is causing full loss of function. So what we did is we performed whole genome sequencing, and indeed after performing it, we found quite a large deletion, more than 4,000 base pairs, encompassing a full exon. And that solved, now we have these compound heterozygous variants, and that solved the case. Second case I wanted to show was another variant in the promoter. So this was a patient already with neonatal disease of leukodystrophy. And whole exome sequencing did not report any candidate variant for this sample. While then RNA revealed for this sample only 2 genes that are aberrantly underexpressed, and the highest one is being UFM1. And UFM1 is a well-characterized gene that is known to cause exactly that disease, the one that the patient had. And then that led to West analysis, and indeed there was found a 3 base pair deletion in the promoter region. And because it's so high, it was discarded. Depending on the place and the workflow, some centers tend to discard variants, even if they're found by whole exome sequencing, tend to discard variants up to certain 100 or base pairs up or downstreams or intronic. So that's why this variant was completely discarded, even though it was detected. And because this same variant was already reported as pathogenic since 2017, the case was solved. So yeah, those were 2 specific samples, but we have many of those. Here in the x-axis, I'm showing the expression fold change with respect to the cohort. So this is the average in one. 50% means is usually indicative of a heterozygous loss of function variant, while a higher reduction is usually indicative of biallelic or homozygous loss of function variants. And indeed, we show in all of these examples, these are the disease causal genes. That's the outlier gene. The gray dots are the background, the whole cohort. And we usually see that in case of heterozygous, we have, it's a bit high, maybe 30 to 40% reduction in the RNA. And at some point, it becomes stronger when we have a biallelic loss of function variants. And that's the previous one was direct, right? You have a loss of function variant in the gene, and that leads directly to underexpression in that same gene. But we have found other very interesting cases of consequences, so of downstream consequences. So it's not the same gene where you have the mutation that is necessarily the one that is aberrantly expressed. So what do I mean here? For example, this case, the causal gene was this gene LRPPRC. And that gene is known to regulate the stability of mature mitochondrial transcripts. And that the disease causal variant was an in-frame deletion, and we see the expression is just slightly reduced, is somewhere there. So it's definitely not an outlier, because and it makes sense because it's just an in-frame deletion, so not out of frame. And indeed, we see all these mitochondrial DNA genes underexpressed because this gene is messed up, and that it disturbs the stability of those transcripts. The same with this other case, LIC3 is a gene known to cause the disease called mitochondrial DNA depletion syndrome. And again, we see many mitochondrial encoded genes underexpressed, including LIC3. LIC3 is also there because it has one stop variant. So in this case, the stop creates the loss of function, is also underexpressed, so unlike the LRPPRC, which is just an in-frame deletion. So two super interesting cases of how through the consequence, we were able to find and or confirm the disease causal effects of these genes. So that was aberrant expression. So now going to aberrant splicing, we saw already with Chris that is a way harder problem because it can take multiple forms. So aberrant expression is usually just the gene is up or down regulated, but with splicing, it can take all these forms because of different variants. For example, a variant here in the acceptor site can cause exon skipping. An exonic one can cause exon truncation. One in the direct splice site can cause exon elongation. One in the intron, super common ones, can activate an exon, or can cause a full intron retention. So again, I'll give some examples. So this one from Sofardi from Epicare, the ERN. It's a female patient that suffers basically from epilepsy. That's what this ERN investigates, epilepsy. DNA through no candidate variants, I think after whole exome sequencing. And then through RNA, in this case in blood, we found 2 exons skipped. So here you can see these 2 exons. They're skipped in 50% of the reads. So sometimes they are taken, but not all the time. So it's very common to do sashimi plots when investigated, but then you're able to see only one case. And what if this alternative splicing is common? And that's why we detect aberrant splicing, not just alternative, in this cohort of more than 250 samples. And then we see that usually the observed Jacquard index is around 100% for all the cohorts, except for this sample of interest for both the donor and the acceptor site. In both donors, so from this side and from this side, then 100 except for the sample of interest, in which is reduced basically 50%. And when it's reduced 50%, it's indicative of something going wrong in just one of the 2 alleles. So through DNA reanalysis, we found indeed a heterozygous deletion here comprising these 2 exons. This was confirmed by qPCR, and it was known to, or discovered that it was transmitted from the father, but it's fine because incomplete penetrance has been reported for this gene, which is a dominant one. Another case is this of a patient that suffers from acute liver failure. Whole exome sequencing didn't reveal anything, so just whole genome sequencing was performed, and that also didn't reveal anything interesting. Maybe the most interesting case was a synonymous variant in this gene twink, because it's a gene known to cause mitochondrial DNA depletion in the brain and the liver, and so it fitted the phenotype of the patient quite well. However, it'll be very difficult to diagnose a patient with a synonymous variant. So RNAseq was performed, and then we saw that exactly on the spot where the synonymous variant is, the exon got truncated. It's homozygous, so we see it's full exon truncation. And that doesn't happen in other samples. So this is just a control. We don't see this truncated part both in the coverage, in the full coverage, or in this sashimi plot. So this variant is activated here, a new splice site, and that leads to an out-of-frame exon truncation, which also caused aberrant expression. So here we see the consequence that was not really expected from this synonymous variant, and that's why it could not have been solved just by the DNA analysis, even though it did capture the variant. Finally, the last case I will show, Chris already touched a bit on this gene TIMDC1, where a new exon was created with respect to other samples where we only see weak splicing. And why do I touch again? Because, not just it's an example of exon creation. So since then, this gene was already published in 2017, since the original publication of Kremer. But other unrelated individuals have been published since with the same variant. And that motivated a group to try to design some treatment. So it's not common to see genetic treatments yet. Basically, what they did is using these oligonucleotides, a correction of this intronic TIMDC1 variant that was reported 5-6 years ago. And the idea is that you fix the missplicing, and because of that, in the cells, and because of that, you can cure the patient or get a treatment for the patient. But not all is perfect with RNA-seq. It's not 100% effective. We saw from the studies that it just uplifts by 8 to 35%. And why is that? Well, first of all, not all genes are expressed. So here we see all protein coding and OMIM genes. Depending on which tissue you take, you get around 60-70% of the genes that are expressed. And that means you cannot test the rest 30-40%. You're completely blind to that. Moreover, we are usually restricted to these clinically accessible tissues, which are blood, muscle, and skin. It's basically impossible to test the brain, which would be probably the most interesting one for example, neurological disorders, liver, heart, and so on. We are simply restricted to those three. Now, even if the gene is expressed, the second problem is that there's these isoforms that are expressed in a tissue-specific manner. For example, this gene, TRPC6, we see that these 3 exons are expressed in the lung, but that's not the case in the brain. Finally, because we are detecting outliers statistically, we need sufficiently high coverage. So if the mean read counts are too low, we simply cannot, we are not able to recover outliers. We see at some point we recover 100% of them if the mean is quite high. But if it's very low, we simply cannot. So if not only the gene is not expressed, but if it's lowly expressed, we cannot detect at least underexpression outliers. So how to overcome this? One way is by predicting. So here we developed this method a couple of years ago, was finally published this year in the lab. The idea is to predict around splicing in any human tissue. So there's 2 or even more well-established tools that are in diagnostic in the labs. Usually SpliceAI is the most common one, but none of those are tissue specific. So what we design is these so-called splice maps, where we say if there's a variant here in the intron, but there's no exon here, like in the brain, so in the stomach and in the heart should be fine, but because there's an exon here, it will impact the brain but not the other 2 organs. That was the main reasoning. So and we were able to develop this using GTEx, so more than 16,000 samples to check the variants and the splicing. The first thing we did is compare a SpliceAI and ME-Splice, and as ground truth, we used the splicing outliers called using Fraser on all the samples, all 16,000 of them, and we see that the precision and the recall are quite low. So by using this, removing underexpressed genes, taking into account and use splice sites, taking into account weak splice sites, plus using the scaling law of splicing that says basically the splicing is always between 0 and 100%, but it matters a lot, which is already the reference. So basically the average across the population, it matters where you are at the start. So, and that influences how much the can filter. So using all of that, we were able to uplift the precision of MSplice and MSpliceAI up to 3-fold. So we are here with this method that we call AB-Splice DNA, way higher curve than the rest, and now it's tissue specific, and it was published a couple of months ago, and it made it to the cover of Nature Genetics. So that's a way to do it in case we want to predict, for example, what will be the consequence in the brain. We cannot test the brain in the patient, but at least we can try to predict using GTEx. So the conclusions of this part, first of all, diagnostics should be interdisciplinary, not just restricted to certain specific fields, but like it's better to work in a team, on my experience. Then RNA-seq can and has increased diagnostic rates by two different ways. The first is validating candidate genes or variants. Then the second avenue is to suggest new genes to be inspected, usually genes that were missed by whatever reason, through whole exome sequencing analysis. Then it has had an 8 to 35% uplift in diagnostic rate. It varies. It's a large variation. Why is this? We think it depends on how you recruited your patients. Sometimes they're already candidate variants, sometimes not. Also, the disease entity and the probe tissue might have a say there, but this is more or less the standard rates of diagnostic. Then we have seen that fibroblasts is the accessible tissue with the most expressed genes with respect to blood and muscle. So one recommendation could be that samples are collected upon the first visit, and that fibroblasts are quite good because they allow for follow-ups experiments, unlike blood. Then we developed AB Splice, which is tissue-specific around splicing prediction, and that can help overcome not probing the desired tissue. And finally, there's potential treatments via splice switching antisense oligonucleotides that are still in the make. Future directions, so where can this technology lead us to, or what are the immediate and short-term and medium-term directions to take? So all of this presentation has been based on what is now called short-read RNA-seq. But long-read RNA-seq is emerging, and it has the potential to quantify isoforms more precisely. So here the question is more like how cost-effective it is, or it will be in the near future. And when that happens, we'll have to adapt all our tools that we have showed. We don't know if they will work as they are for this technology. Then very interesting would be now to integrate with other omics, for example, proteomics or metabolomics, also both statistically and biologically see the impact, how they can be modeled together, for example, or use them together to conclude, to give more strength to a diagnosis. Then also, most of the cases I showed, we saw that it was whole exome sequencing that was failing, but now we have whole genome sequencing that is also becoming cheaper and therefore more accessible. And whole genome enables the study of complex variation. So it's not just big deletions, but also inversions, translocations, duplications, short tandem repeats, and so on. So the question here will be, can RNA-seq capture their consequence? So we'll need to have cases with those different variants, and then coupled with RNA to be able to investigate how good RNA-seq performs there as well. Then regarding the methods, so again, all the cohorts that we have inspected, it's usually in the hundreds of samples. So are the methods ready to cope now that I guess in the very near future, for example, with Genomics England, they already have thousands of samples. In Germany, it will also escalate. So are the methods ready to cope with that, or do they need to be adapted as well? And then for DNA sequencing, there's usually these platforms that allow you to inspect RNA-seq events. But for RNA, there's not such thing. So I believe a browsable platform will be very much useful for this purpose. So with that, I'd like to thank also from Chris' side, well, we both work and studied in the Technical University of Munich, so especially Julian, our PI, and many other colleagues that we've worked together throughout years, developing the algorithms, different algorithms that I presented here, also from the handholds from the lab of Holger Prokic, where we started studying these problems since, I think, 2015. And then we worked together with them in all these mitochondrial disease cohorts. From other institutes, I also showed examples from, in this case, from Barcelona, Japan, and UK. From Solvardi as well, I showed one example from Epicare, but now that's where we have the latest data sets that we're working very hard to get some interesting solved cases. And of course, all the patients, including in the studies and their families and the different funding. And now with that, I'd like also to thank you for attending, and I'll be happy to take any questions from my part or also from Chris' part as well.
Thank you, Chris. Thank you, Vicente, for this really nice update on what RNA-seq can do to support disease diagnostics. And as you said, we're going to start with a Q&A session. So if anyone has a question, please don't be shy. Feel free to unmute yourself and just ask, or put it in the chat. We don't have any yet, so maybe I can start with something a bit more general. So in one of your summary slides, you said that interdisciplinary research is really important. Could you elaborate a bit on that? What the benefits are and what it is that makes it so important?
Yes. So a couple of months ago, we had this event within Solvardi that we call the Solvathon, in which we presented the mathematicians, we presented the results that we had gathered from the hundreds of samples that we collected from the ERNs, the European Reference Networks. And by ourselves, the bioinformaticians, it's very difficult because we don't work with the patient. So we haven't seen the patient, we don't know the exact phenotypes, we don't know the family, so the mode of inheritance, whether the sibling also had it, whether the parents also had it. And we also don't have that knowledge. So working together with the people that did experiments, that collected the samples, and with the clinicians themselves, turned out to be great. Like we diagnosed on spots. We were from the RNAseq group, but there were also people that analyzed the DNA that could then recheck, because you saw that there were many variants that were missed. So they could recheck in whole exome or whole genome sequencing, and then they found the large deletions on the spot that they were simply missed by the standard variant calling. So I believe it's quite good. And also from the other side, because the geneticists, they understand quite well their patients, but they might need this help from the bioinformaticians to interpret these delta psi, these fold changes and everything, to really nail down that it's not just, you can do one sashimi plot, but like you cannot do sashimi plots in one IGV. So you really want to be sure that you have a rare aberrant expression or aberrant splicing event. Like for variants, it's quite easy because you have the Genome reference data set, and you say the variant has to be less than 1 in 1,000. So you're not going to, you don't have to start with 1,000 RNAs. So you really have to understand all the statistics behind this, the p-value, the FDR, and so on. And for that, I think it's best to do it together with the team, to be able also to plot, visualize, and yield a confident diagnosis.
Yeah, makes total sense. Where do you think is it going in the future? Is RNA-seq going to be part of standard care?
Maybe I can take that. So for sure, like currently it's still the DNA, the default, the first part. I think in the end, since RNA and also DNA is very complementary, we need something on the functional assay side. And so if it's RNA proteomics, it's really, I would say, still in the debate, but I think RNA is right now the best way and the most cost-effective thing. So I think it will be more established by the time. But as Vicente said, also like with the interdisciplinarity, we need more training and more expertise and experience basically by the doctors, so that they are more willing to kind of like dive into it and use it. Because currently it's still kind of like a more research side part. So it's more like we need to get it more into a routine that would kind of give also the boost to RNA. But I believe that we need for sure something on the functional side, so proteomics or RNA, to actually help the interpretation of variances.
How do you think that it would be like, how would we be able to combine it with, for example, multiomics, so metabolomics and such?
Vicente, or?
Yeah, I can answer. So one way is, if you see the reduction in the gene and in the corresponding protein, it could be that it's not fully significant. For example, we see the reduction due to NMD, but if it didn't happen fast enough, it cannot be, it could be that it's not fully significant. And then if you also see in the protein, then you have two sources of evidence, or whatever other omic, you have more sources of evidence to assign to diagnose. That can be also especially important if it's a new disease gene that you are trying to characterize. So you really have to be sure of the effect from one side. Then other way would be to see all these consequences. So for example, we see that I showed these two examples where a defect in the RNA had multiple consequences. And that can also be seen at the protein level or at other omics levels, whether these consequences were indeed true, or it could be that for whatever reason, some technicality, the mitochondrial DNA was not expressed in that patient because of experimental or whatever, right? But if we see in another independent layer, then we can be more certain. And one last could be to model it together. So we model the gene expression with a negative binomial and proteomics maybe with a log normal. But what if we say that these are coupled, right? The protein measure with the gene expression measure, they are coupled in a way. It's known to correlate. So it could be that you model them together. We haven't gone on that way yet. And then we have, again, potentially a stronger way of saying, indeed, there is definitely something wrong here because we have both. We have a reduction by modeling together the RNA and the protein, or independently and find a reduction in both of them.
But for example, the clinicians also do PCR to confirm our results. So how would it be like an improvement to also use metabolomics or proteomics? Would maybe improve the diagnostic rate?
Yes. So another thing is, so PCR, you're able to validate one candidate, while in the other approach, you test everything that can be measured. So it can, it gives this independent thing, and it can yield some independent results. For example, I showed this example of a missense variant that by itself it did not have any effect on the RNA, and that's fine. It's expected to, but missense variants do have effect in the protein, so it's even complementary in that sense. So in that sense, they can also offer complementary and not just supporting information.
All right, thank you.
Are there any further questions? If not, then I would like to thank Vicente and Christian again, and thank all of you for listening to this webinar. Have a nice evening, everyone. Hopefully see you next time. Thank you.
Thanks a lot, Nicole, for the invitation.
Most welcome. It was a pleasure to have you here, both of you.